Chapter 23, Serological and Molecular Detection of Viral Infections. So we'll go over what a virus is, the basic structure of it and its life cycle, of course the immune system, what defense mechanisms we have against the viruses, how the viruses escape it, the testing we actually do in the lab for viral infections. So we're going to actually emphasize big time on hepatitis A, B, C, D, and E, certain herpes viruses, Epstein-Barr virus, EBV, CMV was cytomegalovirus, VZV, which is varicella zoster virus, and rubella, mumps, and then HTLV 1 and 2, which is human T-cell lymphotrophic viruses. So here's a basic structure of a virus. Of course, it's very submicroscopic, so we're talking about in nanometers. So you can see the outer envelope consists of glycolipids and proteins, and that's actually derived from the host cell membrane. So it has a core of viral DNA or RNA and a protein coat, which they call the capsid, that's around that, and then the outer envelope. So we're doing just very basic parts of a virus. Mrs. Murray will go into it a lot more depth. So, viruses are obligate intracellular pathogens. In other words, they have to get inside of us for us to be able, for the virus rather, to actually live and replicate, reproduce. So they rely on the host for us human cells, like I said, for replication and survival. So there are freely circulating particles and intracellular particles. So, you're just looking at the basic pictures here of attachment 1 is the actual attachment of the virus, where it attaches to the receptor on the host cell. So now it is actually penetrating, into the host cell, and that's usually by some type of phagocytosis or some other mechanisms. So once it gets in there, it uncoats itself. So the viral capsule degrades, and then it releases the nucleic acid inside of the host cell. So once it gets into the host cell, a transcription of the viral nuclei now occurs. We go into translation of the viral nuclei to produce more viral proteins. So once it produces more viral proteins, they will assemble to produce virions that are intact, and then it will then go and butt off of the host, so lice get through and release its own little babies, its progeny as they call it. So what are some of the immune systems against the viruses? Of course, the same ones we've been talking about. The innate defense mechanisms. We'll look at a picture here in a second. If you actually go in your textbook, you'll see it on page 410, figure 23.3. This is the steps that we'll be actually going through. So for the skin, the innate rather, is the skin and the mucous membranes. Again, we have to recognize the PAMPs on the virus-infected host cells. And we have interferons alpha and beta, which are there to also tell us about viruses. Remember, they're a huge type of a cytokine kind of thing that's out there. The humoral antibody responses. So antibodies will attack only free virus particles. So what do the antibodies actually do? They opsonize them. Sometimes they can actually neutralize them. They can allow complement to fix on them to help destroy them, or they can do ADCC, which is antibody-dependent, cell-mediated cytotoxicity. So then cell-mediated immunity, so interferon gamma this time and interleukin-2 will produce T helper cells. So the host cells that contain the intracellular virus are destroyed by CTLs, which are that's the cytotoxic lymphocytes that we're talking about. So if you look at this figure, this is showing you the different types of immune responses. So if you look at A, that's showing you how interferons will inhibit viral replication. It's amazing what they can do. They inhibit viral replication in the surrounding cells around the infected cell. So interferons will stimulate either the natural killer cells, or can give off cytotoxic T lymphocytes. So that's showing you the two different things that can happen there. So the natural killer cells themselves will destroy virally infected things because they see this MHC that's different now. So because it's releasing virions. So they know something's not quite on, right on there. It's unrestricted. When you talk about the cytotoxic T cells, they can actually bind to the viral antigen on the host cell that's infected with the virus, and actually to be able to destroy them. Now, when you talk about E and F, the here two in the bottom, these are when we're talking about more of a specific B cells that recognize the free virions, that will be starting to penetrate. So when you're talking about coming up into here, you can see how the B cell will differentiate into a type of plasma cell. The plasma cell then liberates antibodies that are specific to the virus, which then can neutralize it, complement fixation, antibody-dependent cytotoxicity, or opsonization, to try to target it to be destroyed. Now, how do the viruses escape this? I mean, they're pretty amazing. As you can see, a lot of them live through this kind of thing. So, mutations, flu virus. I mean, my God, there's a gazillion different, mutations that occur all the time. So, new viral antigens. Or, like hepatitis C is a virus that will block the immune system components. I mean, hepatitis C, it's amazing, excuse me, how it can block interferon-mediated degradation of viral RNA. I mean, it's amazing how it can do that. We can suppress the immune response because CMV, it's able to reduce the expression to class 1. If it does that, then we can't realize that it's different, so we can't attack it. The immune function is altered. Epstein-Barr virus will stimulate polyclonal B-cell activation. Of course, when polyclonal goes on there, polyclonal activation, will cause us to produce, different types of antibodies that don't seem to work quite as well. But what it will do, like HIV, what it actually does is suppress the T helper cells, which we'll talk about in another chapter. And then there's a latent state, and you've probably seen on TV all the time, the varicella zoster shingles kind of thing. It can remain in a latent state for years, and then all of a sudden, pop out, reactivate later in life, like the shingles you hear about all the time. So the laboratory testing we do for viral infections is, again, the antibody titers. We do IgGs and IgMs. We talked about in the previous chapter, where IgM is more of a primary response. IgG is more of a secondary immune response, so that we look at the difference in the titers. So you have to look at the normal ranges, not really normal ranges, but look at the significance of titer. That will determine if it's a infection or not. So from the IgG and IgM titers, we can assess the immune status, because we have to use very specific IgGs to the whatever it is, like measles, mumps, rubella, hepatitis, whatever. Also, molecular testing are coming out that will detect the active infection. It's a great guide to determine if the therapy, the antiviral therapy they're giving, if it's working or not. I mean, HIV was a leader in this, to determine the viral load, they call it, to determine if the therapy they're giving is working. Oh, goodness, excuse me. Okay. So, hepatitis viruses. Alphabet on this. So it's hepatitis A, hepatitis E. You notice those two are up there. HAV, HEV. These are transmitted by fecal oral route, so it's more like a foodborne illness is usually what we talk about there. Ones that are transmitted via parental route is like a bloodborne kind of thing. Hepatitis B, hepatitis D, and hepatitis C. So you can see the initials, HBV, HDV, and HCV. So for all these hepatitis, very generalized symptoms, remember flu-like symptoms. Some people will get pain in the liver area, which is the upper right quadrant. The liver can enlarge. If it does get large and big and really gets inflamed, you can get jaundice. The urine will become dark because we're having tons of bilirubin in it. The feces will be light because all of it's passing in the urine now through the kidneys. And then we will see bilirubins will be elevated and liver enzymes, which you'll learn in Clin Chem. That's AST and the ALT are the two big liver enzymes that we see with that. So, make sure I got this right. Okay, so HAV, hepatitis A virus, is an RNA virus. Like we said, it's fecal oral route. So usually it's food is a big thing. It could be food or water, but so it can produce acute hepatitis in most people. Children really are pretty asymptomatic. There is an HAV infection that's routinely done for all children now. It's actually a formalin activated HAV, but it works extremely well. If somebody has been exposed, they will give them the actual antibodies. That's why they call it the HAV immune globulin. If somebody, if they're get exposed, so it's pre-made, a passive type of immunization. So for an acute infection, what we look for is the IgM is elevated for the HAV. Immunity will be indicated with a total anti-HAV along with a negative IgM. So when they talk about total HAV, that's usually just IgG antibodies is what we're looking at in a particular thing. And you'll actually see it on the lab report. They'll call those IgG. HEV, again, is another type of RNA. And again, it is food, waterborne kind of fecal oral route kind of stuff. So there's 4 different genotypes, 1, 2, 3, and 4. 1 and 2 is usually seen in feces contaminated drinking water. 3 and 4 is an infected pork. That's why you always have to make sure you cook pork well. So 1 and 2 are usually where there's poor sanitation, Africa, Mexico, Asia, some in the Middle East. 3 and 4 can be seen. We've seen it in Japan because they don't cook pork sometimes correctly. Can be seen in North America and, of course, in Europe. So it's pretty asymptomatic, pretty self-limiting infections, except for in immunocompromised people again, and also in pregnant women. So what we look for is if it's an acute infection, the IgM of the HEV, antibodies to HEV, will be there. IgG indicates a past exposure. So, we can look for antibodies, or you can detect actually the virus itself in the blood or the stool during an acute infection. Now, usually what they do is an EIA kind of thing with that, because EIA tests detect antibodies of all the 4 different types of genotypes out there. There is an HEV vaccine that's out there. Remember, IgM is usually detected, very quickly. So it's elevated for a few weeks, and then eventually go away. IgG will reach its peak, but it sort of persists for years. I mean, that's the whole purpose of it. So HEV RNA, they're using real-time PCR to identify it. Seems to work well. They find it in stool samples. So that's one way to detect it. HBV, that's the one that, we come in contact with in the lab. That's the one that you should have had vaccinations to. This is a DNA type of virus that's transmitted with bloodborne or sexual contact, IV drug users, needle sticks, and during the birth process. So this is the one that, we have to worry about for you guys. But most, newborns, I think from 1990 or later, have got immunization for it. It's a routine vaccination that's out there. It's a recombinant type to the hepatitis B surface antigen. So it works extremely well. So acute infection symptoms increase. So the older you get, the more symptoms you get. Now, the person can have a chronic infection. In other words, this lasts 6 months or more. And that occurs in infants and in infected adults. So in other words, the infant gets it congenitally and then it passes on. So with a chronic infection, you can have a chance of having liver cancer, hepatocellular carcinoma, or liver cirrhosis. So all this can be prevented by just simple immunization. Now, if you, have not been immunized, then they can give you shots of hepatitis B immune globulin. In other words, a passive immunity, antibodies pre-made to, against hepatitis B to hold you over until your immunity can kick in. Now, let's look at the serological markers for hepatitis B virus. There's HBs, little s, AG. That's hepatitis B surface antigen. This is actually the outer envelope of the virus. That's why it's called surface. This circulates in virus-like particles in the blood. That's how we determine if you have an active infection. That is what we use in the hepatitis B vaccination. So, you won't detect that if the person's gotten the vaccinations because it's recombinant. So it's not really pathogenic kind of stuff. Now, the hepatitis B E antigen is a core of the hepatitis B virus. This is a marker of active viral replication. So that means there's a high degree of infectivity if the HBe antigen is present. So antibody markers that we look for, they were the antigenic. So the antibody markers we look for hepatitis B is anti-HBC, which means B core. That's what that B stands for, the B, hepatitis B, core, little c, core antigen. So IgM antibodies for hepatitis B core indicates current or recent acute infection. That's what they call the core window. So a total, which is basically IgG, anti-hepatitis B core, indicates a current or a past infection. Okay, so the IgM is acute. IgG can indicate current or past infection. Antibodies to hepatitis BE means you're recovering from hepatitis B. Remember, that's the infectious part, the BE antigen. Antibodies to hepatitis B surface, that's what you get when you have immunity to hepatitis B. That's what you will have from your vaccination. So that's okay if you have that. It's when you have the antigen to hepatitis B surface, HBsAg, that's when it means you have an active infection. So let's look at these in a graphical way. So you can see the incubation is pretty long, 8 to 13 weeks. Acute infection, this is where the antigens appear first. Okay, here's the hepatitis B surface antigen and hepatitis BE antigen. So they occur, early. That's in the symptomatic area, and they decline, like with around 3 months kind of deal. Okay, so that's where the immune system is starting to clear. So you can start seeing how hepatitis BE, hepatitis BE is coming up here. Hepatitis BE and hepatitis B surface, which is this one right here, is starting to climb here. So that occurs during recovery and persists. As you can see, it goes on forever. Now, anti-hepatitis B surface, which is usually, we can see here in this purple one here, that appears during the later part of the symptomatic area here. IgM will decline, but the IgG will persist. So this is the antibodies to hepatitis B core, is what you remember we said was a core window is what we're looking for. So you can see here's the antigens, here's the antibodies, here's the IgM. You can see how the hepatitis B core tails off, and the total was basically just as IgG. As you can see, just goes on for years. Antibodies to hepatitis B surface, antibodies to hepatitis BE. So that's what we're looking for. Now, if the person has a chronic infection, that means the antigen stays there. See the hepatitis B surface antigen and the BE antigen? See how they just stay there. They don't tail off. They just are there. So then you get liver damage, possible hepatocellular carcinoma, that kind of stuff. The IgM for the core goes up and down. IgG persists too. So a chronic infection is where the antigen stays there. You see it for months, if not years. Okay, so it just remains elevated. The antibodies to hepatitis B core stay there too. I mean, they don't tail off, you know. I mean, before, when we looked at the previous one, they did. But here, you know, the production of anti-hepatitis B surface does not occur or is delayed in a chronic infection. All right, so comparing the two. See here, hepatitis B surface, you know, they're getting better. Here, don't see it. See, the B is not even here. That's in a chronic infection. All right, so serological testing. This is a basic EIA enzyme immunoassay kind of method. Antigen at the latex bead here, or, you know, it can be a magnet, can be a fluorescent, you know, chemiluminescent, you know, whatever you want, okay, that we're looking through here. So it binds to it, you know, binds to the antibody, will bind. Here we're adding the one that's got the marker to it, the fluorescence or anything. This is the actual beads here. So then you can have, here's the antigen, the one with the bead, and here's the fluorochrome or the enzyme, whatever it may be. Here they're using a magnet. That's what the beads are, the magnetic in this particular case, and then you're seeing the color or fluorescence there. It's just a serological test. Depends on which one you're doing. You know, if it's going to be a enzyme, fluorescent, magnetic, whatever. You know, whatever you label your immunoassay for. Now, hepatitis D is an RNA virus. And what's unique about D is that it requires hepatitis B. That's why we really don't test for hepatitis D, because if you got hepatitis B, then you got D. So it's transmitted parental or perinatal routes. It's a bloodborne kind of thing. So it is a co-infection with hepatitis B. So, like I say, you know, you can check for the antibodies. It really is not a, you know, big deal because, you know, hepatitis B is what we're looking for. People that are chronic HBV carriers can get a superinfection, get a chronic liver disease, liver failure, and that kind of stuff. So hepatitis B goes with hepatitis B. Okay, HCV, usually it's IV drug use is a big one with this. This is an RNA virus. It's got 7 different type of genotypes. Bloodborne, so contaminated blood, sexual contact, perinatal exposure. Most of the time you're asymptomatic, but can go into chronic liver disease. We see some people have it for years and never have anything. Then we see some people in months all of a sudden get it. So what we can do is do antibodies for IgG for screening and diagnosis. They can actually do qualitative HCV RNA used to confirm if they actually have the HCV virus growing in them. So that's what we do to determine if what's called viral load, to determine if they need antiviral therapy. And it's good to find out which genotype it is. But they do now have a great medication for it. It's like a 95% cure rate, so it's under control. But the biggest thing is finding out if the person has it, so we can test them and treat them. Okay, EBV, Epstein-Barr virus. This is a DNA herpes virus. You know, the kissing virus, they call it. Salivary secretions with intimate contact, because it begins in the oropharynx. The B cells and epithelial cells spread it through the lymphatic system. So the usual diseases that come with this is infectious mono, lymphoproliferative disorders, and certain malignancies is Burkitt's lymphoma, which is a real bad thing to have. So most people are infected. It's like 90% of the population have had this. You know, it's just a self-limiting a lot of times. It can be late and it can reactivate, but it's usually kept in check. You know, again, the immunocompromised kind of deal that we're talking about. So for infectious mono, you know, the primary EBV infection, you know, in teenagers that we're looking for is fever, swollen lymph nodes, sore throat, and fatigue. And then the biggest thing is looking for lymphocytosis, absolute, and looking for a reactive or atypical lymphocytes, which you can see a picture of those there. Most of the time they call them cytotoxic lymphocytes. We think that's what the reactive or atypical lymphocytes are. So the testing they can do is called a heterophile antibody. So what it does is that when somebody has it, the antibodies seem to react to different species, like horse red cells, bovine red cells, sheep red cells. We seem to see agglutination with that. There's actual a monospot test that's out there that looks just for that. And it can be done as a rapid, EIA kind of test, and we'll actually do that for our serological testing. The Paul Bonnell test, it's just an older method. What they do is they actually mix the patient's serum with a suspension of sheet red blood cells, and what they're trying to do is sort of detect it that way. As you can see, it's a heterophile. It's not giving us a good result as to what we're looking for. So it's better to actually do testing, antibody testing for the EBV antigens themselves. So talking about the antibodies to EBV antigens, there's early antigens. There's what's called the EAD, which is seen in acute infectious mono. The EAR, which is really restricted to more of the cytoplasm kind of thing. So the antibodies to EA, I don't know why they call it early antigen, but whatever. EA IgG appears in the acute phase and usually falls off quickly, 3 or 4 months. But if you have that, that means it's an active infection, obviously, because it's IgG. But a lot of people will still have this for many years. When you're looking for the late antigens, then you're talking about the viral capsid antigens, the VCA. That's viral capsid antigens. So there's IgM, and IgG for those. So the IgM VCA antibody is the best marker for acute. The IgG will indicate acute or a past infection. And, it's just like with any other antibody titer. Now, the latent phase is called the EBV nuclear antigens. Don't you love all these initials? Epstein-Barr virus nuclear antigens. So they have the Epstein-Barr nuclear antigens too. So the Epstein-Barr nuclear antigens appear, convalescent and slowly appear in onset of symptoms, and they can persist for later in life. So what they usually look for is the VCA a lot of times to determine what's going on with the patient. So antibodies to EBV antigens, like I said, the anti-VCA, if you remember, VCA, and we're talking about the viral capsid antigens. Okay, the viral capsid, means they can have a recent infection. Past infections will be more the IgG kind of stuff, is what we're looking for. Okay. So any higher elevated antibodies. Now, how do you treat it? Well, you let the person rest, because you really, one of the side effects of this is severe liver issues. So you want to make sure that the person rests well. If they rest well, then they more than likely won't be getting, liver issues with it. So that's very, important that the person rest. So acute and convalescent specimens, that kind of stuff we got to look for. CMV is cytomegalovirus. Cytomegalovirus is a DNA herpes kind of virus, and again, it's oral secretions, and you can see it's genital secretions, or it can be congenitally transmitted or transplantation. A lot of the blood products, we actually test for CMV because they can be transmitted in the blood products, so we have to be careful. So it's the same kind of symptoms as mono. Most people are asymptomatic. Over 90% of the population have had CMV, don't even know it. So it's only people that are truly immunosuppressed that we actually have to worry about that. So patients with AIDS, transplant patients, people on chemotherapy, that kind of stuff. So it can cause problems in those individuals. Can cause life-threatening issues. There can be congenital things that go on it. So this virus, is latent. It can stay in monocytes in this particular case, as opposed to lymphocytes in EBV. And it can be reactivated. So it has to be very careful with that. So to diagnose it in the laboratory, you can do cultures for it. We can do molecular testing, looking for the actual antigen and looking for the DNA also. Serological testing, we do that to screen all blood donors and organ donors. So if you look for IgG, antibodies to CMV will indicate an infection. So we got to be careful with that. So a viral infection, the viral culture rather, it's a tough thing for it to grow. So a lot of times we just do serological testing with it. It can be a fluorescent testing. It can be enzyme-linked immunoassay. So there's multiple ways that it can be done. They do have molecular testing that's out there now, quantitative PCR, to determine if the patient has it or not. Okay, VZV, varicella zoster virus. It's a DNA herpes virus. It's transmitted by infected respiratory droplets. You inhale them. That can come from the skin lesions. So chicken pox, which they have vaccinations for that now, and shingles, which they do have a vaccination for that. It's a 2-shot deal now. Very preventable. Chicken pox is, a lot of kids used to get it. When I was a kid growing up, for some reason, they used to have chicken pox parties, which now sounds awful. But every kid would get the chicken pox. You get all these little itchy blisters over your body, and it would eventually go away. But the virus, it can be latent, and it can travel in the sensory neurons and then become reactivated later, and that would be the shingles. And from what I'm hearing, shingles is extremely painful. It's like getting a hot poker sticking it. It burns, a lot of debilitating pain. It's awful. So vaccination is very important for this. So to diagnose it, you just go by usually the clinical findings. You can do real-time PCR. Serological methods are used, but the reason we only really do that is to make sure that people have immunity to it. So if you're dealing in people that have shingles or have chickenpox, that you have immunity so you don't get it. Also, like in organ transplant patients. So that's something that, we're going to have to check on you before you actually do your clinicals. Rubella is an RNA virus. It's transmitted again through respiratory droplets or across the placenta. This is actually German measles. Used to be a huge thing when I was a kid, German measles. It can cause genital abnormalities, miscarriages, stillbirths, easy infection, immunization for this particular thing. It's very self-limiting. Yeah, they get swollen glands. Sometimes you get a skin rash with it. But, it used to cause a lot of congenital abnormalities, where the deafness, cardiac problems, mental retardation, it used to be a big deal. So now that we've got a attenuated live virus vaccination, works really well. That's usually the MMR shot you hear. But it's just measles, mumps, rubella. It's the normal vaccination that we give to everybody now. So again, immunization kind of stuff, look for IgG and IgM to determine what's going on. IgG means immunity. Congenital infection, we can find IgM antibodies in the fetus. That means the baby's now got it. So you have to be careful with that. So you can do viral cultures. Molecular methods is an RT-PCR kind of thing for this. Rubeola is a little bit different. Rubeola, again, is considered like a type of measles. They get a skin rash is usually the biggest thing that we'll see in this particular one. It can cause a subacute sclerosing panencephalitis. So you can see in this particular kid how he's got the rash, all over his body here. Again, it's an immunization kind of thing. You can do serological test. Look at the fourfold rise of acute and convalescent, looking at IgG and IgM titers to determine what's going on. Again, it's part of the MMR shot. Mumps, RNA virus this time. You can see how the product glands are really swollen. So it's transmitted again through respiratory droplets or saliva. Again, it's preventable by immunization. Again, you can do RT-PCR, but usually it's just clinical presentation is how we do a diagnosis of it. So it doesn't really come to us that much at all. Okay, HTLV1 and 2. This is human T lymphotrophic virus 1 and 2. It's a retrovirus. This is how, we first thought this is what HIV actually was, but we found out it wasn't. So it's transmitted usually by bloodborne kind of stuff, or sexual contact, or perinatal, and then breastfeeding. So HTLV1 causes adult T-cell leukemia lymphoma. That's ATL. HAM TSP is another way that it can actually just be spelled out. So serological tests for antibodies to HTLV 1 and 2 to diagnose it. We screen all blood donors for this. The typical method that is done is ELISA method, enzyme-linked immunosorbent assay. If that's positive, then you can do Western blot. All right, so let's summarize this stuff. So viruses are obligate intracellular pathogens that cause many, many diseases. And what we look for is the antibodies serologically, to bind the free virions so we can stop the spread of infection. Also, cytotoxic T cells or natural killer cells can attack intracellular viruses, because it sees the MHC on them and destroys it. But viruses do learn to escape, the host defense mechanisms. And they have that latent stage. We talked about that with quite a few. Viral infections, we can culture it, we can check for the antigen, we can check for the antibodies, we can do molecular testing, looking for certain nucleic acids to determine if they have it. Serological tests are great. We can do IgM to indicate a current or recent infection or possibly congenital infection. Presence of IgG can tell us about previous exposure and if the person is immune to it or not. That's why it's so important to do both IgG and IgM to find out the immune status of whatever it is we're looking for. Serology plays a key role in a lot of things, especially, when we went through all the hepatitis, HAV, AV, hepatitis A, B, C, D, and E. Okay? A and V is fecal oral, B, C, and D is parentally. Okay, Epstein-Barr virus causes infectious mono, can go into malignancies. There are serological tests for it. There's the heterophile stuff, but the best way to do it is the Epstein-Barr virus specific antibodies to determine what's going on. CMV is a herpes because it causes congenital and really can cause problem in immunocompromised people. Again, serological testing, molecular testing is the best way to go for that. So, many viral infections now, just routine immunization, makes it preventable. And we can identify if the person is immune or not by just doing serological testing, doing IgG and IgM titers. Varicella, chickenpox and shingles, rubella, German measles, rubeola, measles, mumps, HTLV1 and 2. HTLV1 is adult T-cell leukemia. So, when you're looking at all this, it's important to know, viruses, can invade, titers can tell us a lot of information that we need to know, and all the different hepatitis, so make sure you look at those charts. So on page 429, 430, and 423 is some summaries and study guides, and of course the review questions at the end of the chapter. Make sure you review it all. If you have any questions, please contact me. Thank you.